My lab studies the molecular basis of human diseases, and many years ago we realized that many human diseases are due to frame shift or nonsense mutations that result in the premature termination of translation. And we realized that when translation terminates sufficiently prematurely, we figured out the rules for what I mean by that, then the mRNA is degraded. And it's an important pathway because truncated proteins can be toxic to cells. So this is a type of quality control pathway that eliminates mRNAs that have the potential to encode truncated toxic proteins. And then it turns out this pathway eliminates mistakes that we make all of the time when we express our genes because of mistakes made, for example, during alternative splicing. And then the cell uses this pathway to constructively develop autoregulatory loops for RNA binding proteins like splicing factors. And on top of that, this pathway is regulated by cells so that they can respond appropriately to particular stimuli. So 10% of our normal natural mRNAs are NMD targets, nonsense-mediated mRNA decay, NMD targets. So we study all of that and then have gotten into other pathways like Stauffen-mediated mRNA decay and microRNA-mediated decay, because oftentimes proteins multitask in cells, and the key NMD factor, UPF1, doesn't just do NMD, it does other things that we've studied. You know, I've always loved studying RNA, and in fact, fortuitously or serendipitously, I have always studied RNA. Even as an undergraduate, I studied the translation of myosin heavy chain mRNA. And then as a graduate student, I studied RNA synthesis by the lactose operon in E. coli. And then I started to study human diseases, and it's just where our research took us. Receiving this award is an amazing honor. It's an amazing privilege. I do take mentoring other scientists, especially younger scientists, very seriously. I wish I had had more mentoring when I was a graduate student, for example. I was a graduate student in the mid-70s where in my department in biochemistry we didn't have any female faculty members, and I do think there's a big difference between how women approach things and men approach things. So I'm particularly interested in making sure that women develop the skills and the confidence that they need to be a successful scientist. I think science is a wonderful career.